Okay, you are. Okay, so our next speaker is Khalid Machaca. Khalid got his bachelor degree from American University of Beirut in 1990, and then he went to University of Georgia, where he got his master degree, and then from there he went to Emory University for his PhD. After his PhD in postdoctoral work, he set up his lab in Arkansas Medical School, where he spent several years, and that's when I came to know about his work when I joined Peter Young's lab as a graduate student. He gave me, as my second project during my PhD, he gave me a paper which was published by Khalid and his students on maturation of oocytes and how the calcium signaling changes when the oocytes mature. And at the time, I thought it was really, really nice and really clean data. And I really enjoyed working on that project, modeling the data. So that led to two papers during my PhD, but I have been following Khalid's work for, since I, got into the calcium signaling field. And he from Arkansas, after a while, he moved to Doha. And his, you kind of hear these stereotypes that you can do good science in the US and in Europe, and Khalid's story is completely opposite to that. His science actually it went completely to an up, to a different level. When he moved to Qatar, he is doing extremely interesting science there, and the quality of his publications and experiments have gone exponentially up. So I'm really excited to hear about what he has done recently, and looking forward to his talk. So for that, I'll hand it over to Khalid.
Thank you, Ghanem, for a very generous introduction and very kind words. And thank you, Ata and Ghanem, for organizing this. It's really exciting and fun to be able to talk science after a year being in quarantine. So what I'd like to talk about today is a story we've been working on for a few years now. Which is, in my mind, a pretty novel way of calcium signaling that we call calcium tunneling or calcium teleporting. And I hope through this talk you'll kind of get a feel why we call it that. But we strongly believe that this kind of expands the dimensions of calcium signaling, allowing it to specify more both in the spatial temporal direction to ultimately lead to the cellular functions that calcium is encoding. So I don't need to impress much on this audience that calcium has a very broad spatial temporal dynamics. So at the smallest spatial scale, if you will, elementary calcium release events, like the ones we've heard on from Suhita just now, induce vesicle release. This could be due to the opening of a few calcium channels at the cell membrane that generates a very localized calcium rise that induce cellular function, whether it's vesicle release, whether it's activation of membrane channel, whether regulation of metabolism at the level of the mitochondria, or even transcription very locally. So we think of those as localized elementary events. Calcium can also generate global calcium signals at the cellular level, either in the form of a global rise or a sweeping calcium wave that can spread throughout the entire cell, mediating different functions like fertilization, muscle contraction, liver metabolism, so on and so forth. And also organs are set up to do tissue-wide calcium signals through cell-cell interactions, either through electrical coupling through gap junctions or through release of agents like ATP and others that can transmit the calcium signal from one cell to another. So basically, We can go from the nanometer scale to the centimeter scale in terms of calcium signaling, thus encompassing several orders of magnitudes. The same story we can see at the temporal level with a broader order of magnitude, actually, where calcium can signal from the microsecond scale to up to hours, like in fertilization with everything in between. So that brings a very important conceptual question: is with calcium signals being so broad, spatially and temporally, even in the same cell sometimes, how is specificity encoded? How can calcium signal tell the cell, do this or do that, transcribe, secrete, proliferate, move, or die? And calcium encodes all of these things based on these spatial temporal dynamics. So somehow this information must be decoded into a signal. So I like to put up this video that kind of illustrates this in my mind. What you're looking at here is a frog oocyte, which is one of the model systems we work on. And the cell has been loaded with a calcium dye and caged IP3. IP3 is an agent that releases calcium from intracellular stores. And then all I did here is I basically, with a laser, uncaged calcium in this pattern. So if we look at this, for your brain, this makes sense. You can read calcium, it's a signal, you can immediately think it's the coolest ion, it's a beautiful signaling molecule. So, and this is exactly what the cell does. It decodes the spatiotemporal information encoded in the calcium signal into information that then it decides what to do with in terms of a cellular response. So several years back, we've asked ourselves the question. So we know from the literature that calcium can signal very locally through these elementary events. And we know it can produce global events, which is encoding information at the broad cellular level. But can calcium signal in the mid-range? And if it can, how does it do it? And how does it encode specificity? So, I mean, the problem is illustrated in this cartoon here. If you want to activate an effector, a calcium-dependent effector, that is physically quite close to the calcium channel, it's very simple conceptually to do that. You just put the effector close to the calcium channel, the channel opens, calcium flows, the effector is activated, and then you have specificity. Only this effector is activated because of its physical proximity to the channel, and no other calcium effectors in this cell are activated. But what if you want to activate an effector that's further out from the channels? Well, you could say it's simple. All you have to do is either open the channel for longer or open more channels, which gives you a broader calcium signal. And now you can activate effectors that are further out. Now, the problem with that model is the calcium channels on the ER membrane, IP3 receptors or randine receptors, their activity is calcium dependent. So if you release calcium into a broader domain, you're likely to induce calcium-induced calcium release, which then you end up with a global signal. So can the cell specifically activate effectors in the mid-range or not? Or is there really a binary signal between very localized signals and global signals? So we've been working for years on calcium signaling differentiation in the frog oocyte, like Anna mentioned at the beginning in his introduction. And to cut a long story short, if we quickly review the calcium signaling machinery in the oocyte, you have a calcium release channel on the ER membrane that releases calcium from the intracellular calcium store in the ER. The Circa pump puts calcium back in the endoplasmic reticulum. If calcium stores are depleted, that activates store-operated calcium entry, where the calcium sensor in the ER membrane, STIM1, translocates to the ER plasma membrane junctions, binds to and activates ORI, which is a calcium selective channel at the plasma membrane that induces calcium influx. The cell has calcium activated chloride channels that are important for its physiology, primarily mediating the block to polyspermy at fertilization. And then there's a calcium pump at the cell membrane that pumps calcium outside the cell. So combined, if you take this machinery, and if you look at this video here, what we've done here, we've taken the same model and we've uncaged IP3 very locally in this region. So with the entire machinery active, you get calcium released from stores, and then the signal fades over time. This is in the immature oocyte. As the cell gets ready for fertilization, it's maturing, its calcium signaling machinery remodels. Interestingly, this species, the Xenopus laevis, is a frog that does fertilization in pond water. Where the extracellular environment is not well controlled. So the oocyte pretty much removes all the signaling machinery except for the chloride channel from the cell membrane. It internalizes PMCA and ORI into intracellular vesicles. IP3 receptors cluster and mediate much stronger calcium release from stores. And STIM1 can no longer interact with ORI. So store-operated calcium entry is completely inhibited. Not much happened to circa. Now, what's really fascinating is when you change all of these things physiologically, now the cell becomes electrically excitable, and that initial transient that we induce here with the release of IP3 leads to a broad calcium wave that sweeps through to the entire cell. And this is the change that Ghanem modeled in our collaborative work early on. And what's fascinating is if you plug all these physiological changes in a mathematical model, you replicate all the changes we see globally and even at the single events, the elementary events with TAMP, which I'm not showing you here because of time. So we feel with this that we have a pretty good understanding of the changes happening during oocyte maturation and calcium signaling remodeling. But what I want to focus on today is more store-operated calcium entry. And as I mentioned briefly, STIM1 is a transmembrane protein that localizes to the ER. It has EF hands in the lumen that bind calcium. When there's a signal, an agonist-dependent calcium release through GPCRs or tyrosine kinase coupled receptors, STIM1 loses calcium in the EF hand because of the decrease of calcium in the ER. It clusters, it moves to the ER plasma membrane junctions, it traps ORI by diffusional coupling, and then once it binds to ORI, it gates the channel open, allowing calcium influx. So we've used over the years these calcium activated chloride channels in the oocyte because they're beautiful markers for changes of calcium in the cytosol. And we've characterized very carefully these changes as dependent on either calcium release or calcium influx. So let me walk you briefly through this. The methods we're using is a 2-electrode voltage clamp to control voltage across the membrane of the oocyte and recurrent current. At the same time, we can have a 3rd electrode where we can inject IP3, gauge calcium, whatever we want. So if you do this and you use the simple voltage protocol where you clamp the cell at +40, then -140, then +40 again, at rest, there's basically no activity. If we induce calcium release from stores, we see the calcium activated chloride channel being activated. We call this current ICL1, and you see a sustained chloride current at both +40 pulses because calcium is high in the cytosol here or here. After the stores are completely depleted, now calcium is flowing into the cell from outside. There's no more calcium being released from the stores. And when this happens at the first +40 pulse, because calcium is low in the cytosol, there's no activity of the chloride channel. If we step the cell to minus 140, we're going to increase the driving force for calcium influx. Calcium flows, activates the chloride channel, and you see the chloride current being activated. And then if we step to plus 40, that calcium activates the chloride current in a transient fashion, because at plus 40, we're inhibiting calcium influx and calcium is disappearing. So we've used typically the amplitude of ICLT as our measure for store-operated calcium entry, and the amplitude of ICL1 as our measure for calcium release from stores. What this allows us to do is to separate calcium release and calcium influx in real time in the cell as we're studying it, without affecting calcium signals in any way by injecting a calcium dye to look at calcium changes or anything like that. We're using an endogenous effector to look at these calcium changes. So the initial observation that got us into this, and we scratched our heads for a long time when we saw this, is an experiment that Raphael did in the lab, where we were just looking at calcium dynamics in the oocyte. And if we empty the stores with ionomycin, which is a calcium ionophore that preferentially inserts itself in the ER membrane and releases calcium stores, we activate calcium influx, we activate store-operated calcium entry. I mean, you don't see it in this scale, but there is calcium influx at activation of ICLT under these conditions. But the striking observation happens is when we injected the cell with IP3 to empty the stores, now the amplitude of ICLT was orders of magnitudes higher, and this is quantified here. So this was really confusing. You know, we're emptying the stores in the same way, we're activating store-operated calcium entry, yet somehow, based on the method by which we're emptying the stores, we have the same current activated to much different levels. So we tried different ways. We emptied the stores using T-pen, which is a heavy metal chelator, has a low KD, a high KD for calcium, and is membrane permeant, so it goes into the ER, binds calcium. And although calcium physically never leaves the ER because it's bound to T-pen, it's as if you've emptied the stores. That does activate ICLT better than ionomycin, but similar broad range. Or we can block the Circa pump using thapsigargan. This is an irreversible blocker. Because there's a continuous leak from the ER, the stores will be emptied, SOC will be activated, and you activate ICLT. That also activates it, but nothing close to activation with IP3. So the first thing we thought about is, well, what if, depending on the methods of store depletion, somehow this induces more calcium influx through store-operated calcium entry? So we tested that directly now by measuring calcium influx through SOC, not by using ICLT as a marker, but directly measuring calcium entry into the cell using 2-electrode voltage clamp. And here you see the typical inward rectifying current of store-operated calcium entry. And this is the current we get with ionomycin. This is the current we get with IP3. They're not statistically different. And certainly they cannot explain this huge differential activation of ICLT. So IP3 is activating this effector through a calcium-dependent pathway, not through increasing the amount of calcium flowing inside the cell. And we did the same thing by voltage clamping the cell now, but at the same time imaging calcium inside the oocyte using a calcium dye. And at plus 40, this is the basal fluorescence of the dye, reporting basal calcium level. We go to minus 140, we see an increase due to calcium influx from outside of the cell because we've activated store-operated calcium entry. and then we go back to plus 40 and the signal goes back to baseline. And if we quantify this, again, something quite interesting emerges. In control, there's no signal, ionomycin, we do get calcium influx. And in most of these cases, as you would expect, the calcium flow is cortical. Remember, this is a huge cell, it's 1.2 millimeter in diameter. The frog oocyte is quite large. So we can actually quite well see the cortical calcium signal. If we now empty the stores with thapsigargan, you see a robust calcium signal that's close to that we see with IP3. Because the calcium flowing can no longer be taken up into the ER store, it lags more in the cortex of the cell. But for some reason, this calcium is not effective at activating ICLT. Compare the ability of the activation of ICLT here with the calcium transient we see cortically compared to IP3. For some reason, this mode of calcium rise, although just broadly looking at the level of calcium in the cortex, I mean, they're statistically different, but they're of the same order of magnitude, somehow leads to a very different activation of an effector in that region, which is quite fascinating. So to guide you through the rest of the data, I'm going to actually put up the model that we came up with, and I'm hoping this will help you better follow the data. So this is what we think is happening. You empty the stores, STIM1 clusters at these ER plasma membrane junctions, it binds to ORI, activates calcium influx. Because ORI is a low conductance calcium channel, that calcium signal we think is pretty localized to these junctional complexes. SERCA localizes to these junctions and prevents calcium from spreading out of the microdomain by taking it back into the ER. Now, typically what would happen if the signal is terminated, that would refill the stores, dissociate STIM ORI, and you go back to rest. But under conditions where the IP3 receptor is open, as would happen under the conditions we've created where we've injected IP3, now you have a leak pathway that this calcium coming into the ER leaks out of the ER through open IP3 receptors and activates these chloride channels, the NO1 channels, which are the calcium activated chloride channels. Because the conductance of the IP3 receptor is very large, a single IP3 receptor could release calcium flowing through many SERCAs into the ER. So what's the evidence for this model? So again, let's take our favorite effector on the cell membrane that's activated either by calcium release from stores to generate this current ICL1, or calcium influx through store-operated calcium entry to activate ICLT. And this is a typical experiment where now we inject IP3 into the cell. You see calcium being released from stores as reported by ICL1. And then as the stores empty, that current dies down. As the stores empty, store-operated calcium entry is activated, ICLT is activated, and then it peaks. Now at that point, when the stores are empty and SOC is fully activated, if we inject heparin, which is a blocker of the IP3 receptor, ICLT current dies down. So this tells us that a functional IP3 receptor is required for the high ICLT current we see following IP3 injection. We can do the experiment in a slightly different way. Remember, I showed you the two extremes with ionomycin activating ICLT, but at low levels. Same experiment, we treat the cells with ionomycin, it dumps calcium from the stores, you activate ICL1, now with much more rapid kinetics as you would expect, the stores are empty, and you can start
Khaled,
seeing.
can I
Yes,
just ask you to go back to the model because I like to
sure.
understand the model better before we go into more data. So, all right. So I think I've got the outline of the model, but I was kind of curious as to the numbers of these receptors, the aura one. Like when you say local, um, in order to get a better understanding of what local means, can you maybe give us a better idea of the spatial density of these receptors, the average separation, say, of the aura versus the the IP. Well, I mean the IP3 you're injecting, so let's not worry. But the endogenous ones, which is the aura one and the ano one. So what
Yeah.
are the sort of numbers for in a oocyte for endogenous like chloride channels and
Right. So I'll show you in a few minutes the spatial distribution of these effectors under the
okay,
different conditions.
okay,
Now, in terms of absolute numbers of ORI channel and a one channel in the oocyte, all we can rely on is the amplitude of the currents. And working back using the single channel conductance of ORI, NO1, we can do the same thing. The important thing in this model is none of these is changing during the course of the experiment. This is the same cell under the same condition that we can activate differentially between the two. So yes, I agree with you. It's very important to know the numbers and their spatial distribution. But it really wouldn't explain the differential response to ionomycin and IP3.
well, I mean, and if you do have the unitary conductances, which you seem to do, then you can actually make estimates. So I mean, so
Correct.
that helps. So
Correct.
it helps to know that, and then if you've done spatial distribution imaging to look at clustering and so on, that's great. Okay,
Right.
got
Right.
it.
And I'll show you the estimated spatial separation between store operated calcium entry and the chloride channels actually. And it's in a little bit, just a couple of minutes. So again, if we empty the stores with IP3 and we block the IP3 receptor, this calcium is no longer as effective in activating ICLT. Although, as I've shown you, the amount of calcium flowing through SOG is still there, but it activates very little ICLT. Now, if we do the experiment differently, we empty the stores completely with ionomycin. So no IP3 activation here. We just put holes into the membrane or put an ionophore here that releases calcium from the ER. We empty the stores. We activate store operated calcium entry. You see it rising ever so slightly here by activating ICLT. And now at this point, after the stores are empty, if we inject IP3 to activate the IP3 receptor, boom, you see a huge activation of ICLT. So somehow when we open this channel, that calcium flowing in is now tunneled through the ER, enriched in a way where it's much more effective in activating ICLT. Although the amount of calcium flowing into the cell through SOG here and here is identical or very similar, right? So simply by activating these different channels in different ways, the amplitude of your effector is orders of magnitudes higher. So this is in response to your question, Shahid. So what we've done here, we've tagged the chloride channel, NO1, which is the calcium activated chloride channel, with mCherry molecularly. And we've tagged STIM1 back to the model. So this guy is green now, and this guy is red, right? And we're asking what happens after store depletion. And what's really interesting is there's almost well, not almost, there is exclusion of these two microdomains. So STIM1 forms clusters at ER plasma membrane junction. And just to put this in context, this is a frog oocyte. So everything is, you know, much larger than you would see in a mammalian cell, but conceptually the same thing happened. And that's why we like this model, because it allows us to spatially see things that would be very difficult to see in a typical mammalian cell. But you see STIM1 goes and forms these puncta and almost excludes the chloride channel from these puncta. So I'm not showing you these images, you can go to the paper, but NO1 at rest is diffuse throughout the whole membrane. And STIM1 is not close to the membrane because it's in the ER. But once you cluster them, STIM1 goes to these ER plasma membrane junction, and NO1 is no longer there. The chloride channel is no longer there. So the calcium flowing here is somehow tunneled to this microdomain to activate it. And we've estimated the separation of these distances. A typical STIM ORI cluster is about 0.6 micron, and the distance between two separate STIM clusters is about 2.6 microns. Now, for the model to work, CIRCA needs to co-localize with STIM in these domains. So here we've tagged CIRCA 2B with GFB, STIM1 with cyan, and ORI with cherry. And if we empty the stores, you see all three co-localize in these ER plasma membrane junctions close to the cell membrane. And this is an orthogonal section through a Z-stack of images. So if we're taking a confocal section, we're crossing through the oocyte, and then if we optically cut across, you can see a cross section of the cell membrane. You see the junctions, the ER plasma membrane junctions there. Although there's still some CIRCA deep into the ER, certainly 1% of the CIRCA localizes to these ER plasma membrane junction, and this is a 3D here. So the way I'm showing these images is 2 at a time, because with all 3 it becomes very confusing, right? So CIRCA 2B co-localizes with STIM1, it co-localizes with ORI, and of course STIM and ORI co-localize as we know happens. So again, back to the model, CIRCA does co-localize to these ER plasma membrane junction where store operated calcium entry happens, and one would expect it would take calcium into the ER. And with an open IP3 receptor now and a separate microdomain, as these two domains are actually physically separate, it allows taking this calcium, concentrating it, tunneling it, and releasing it here to activate this channel much more effectively than would be possible if this calcium flowing in is directly activating NO. So again, this is the model. Calcium flowing through the STIM ORI microdomain is taken up into the ER, re-released, now much closer to the chloride channel, inducing much more effective activation of this effector. So what that kind of implies is that the ability of the machinery to take calcium from the microdomain and re-release it now is able to activate, as I've shown you, an effector that's at the cell membrane. But what happens to an effector that's deep inside the cell? Could this tunneling work in the same way to bring calcium through the ER to activate a much deeper effector? So Peterson and colleagues many years back have shown that this actually does happen in acinar cells, in pancreatic acinar cells. And the way they've done these experiments is they put the cell in calcium-free media, they patch on the cell with a calcium containing solution, they activate store-operated calcium entry. In the old days, it was called capacitative calcium entry. It's the same channel. And they can show that this calcium flowing through calcium in the patch pipette can activate zymogen granule release on the apical side of the cell if they do it basolaterally. And they've done some more work to show that the ER is pretty continuous luminally across the cell, so it's able to tunnel calcium from here to the other end of the cell. Now these cells are pretty specialized, and they're secretory cell. That's their job in life. So we wanted to ask the question: does this happen in a regular fibroblast or a regular cell? And we chose HeLa cells for these experiments for the main reason that the mitochondria in these cells are quite deep inside the cell. And because the mitochondria are heavily dependent on calcium signaling, it seemed to us a perfect model to ask the question whether calcium tunneling can affect or can activate effectors that are deeper in the cell, in addition to the effectors at the cell membrane, like I've shown you for the chloride gel. And the way we went about doing these experiments is we wanted to image calcium in the cytosol, and we used Fura red for that. We also wanted to image calcium in the ER lumen and in the mitochondria in real time. And for that, we used genetically encoded calcium reporters, the sepia reporters, and you can target those to the ER. This is the red sepia ER. You can see its ER reticular distribution. In the same cell, we've transfected green sepia mitochondria, and it reports mitochondrial calcium nicely. And you see the separation between the mitochondria and the ER at these high magnification. So if we take these cells, load them with Fura red, now we can image the three compartments in real time in the same cell. So what kind of signals do we get? These are just setup experiments just to show you that it works. So if we take the cells, we put them in calcium-free media, and we treat them with an agonist, histamine in this case. That results in calcium release from stores. You see, this is cytosolic calcium. So remember the colors. Magenta is cytosolic, red is ER, and green is mitochondria. And the way I'm reporting these, because they tend to be quite complicated quickly, is the quantification is where the bar is, which is color-coded. So if we release calcium from stores using histamine and agonist, you see calcium being released, and you see it's due to depletion of calcium from the ER, as reported by sepia ER, and we can quantify that signal. Now, if we treat the cells with thapsigargan, so this is under these conditions. We activate a receptor, calcium is released through the IP3 receptor, and we see a large cytosolic calcium rise, as you see it here, and we see a decrease of calcium in the ER. If we treat the cells with thapsigargan, as I mentioned earlier, because of a leak pathway from the ER, we see a calcium rise in the cytosol, tends to be slower than with histamine, but the amplitude is not that different ultimately. This is coupled to a much more dramatic emptying of the ER, as you would expect, because the ER can no longer refill with calcium, and that depletion stays for the duration of the experiment, because now the ER has no way to take calcium back. And now, if we give the cell calcium, you see the activation of a huge store-operated calcium entry that's quantified, right? So the system is behaving as we would expect. Now, just to put this in context, the literature, the favorite approach to measure store-operated calcium entry is through thapsigargan, because you can spatially separate calcium release from calcium influx and then study SOC separately. But if we look at what I call physiological SOC, and here we're emptying the stores with CPA, which is also a blocker of the SERCA pump like thapsigargan. But thapsigargan is irreversible. CPA you can wash up. So we treat the cells with CPA, see a calcium rise in the cytosol, the ER depletes, calcium is low in the ER. We just add histamine here to convince ourselves that the stores are empty, and indeed they are. There's a small blip, no change in the ER. But here we're washing CPA out. So now, during that period, SERCA is active again. It's no longer inhibited. Now, if we give the cell calcium, we see beautiful refilling of the ER, knowing that SERCA is working, putting calcium back into the ER. But the amount of calcium in the cytosol is minute. And this goes back to the idea that this calcium flowing through store-operated calcium entry is really not being reported in the bulk cytosol. It's very localized to these microdomains. And you see very little spillage, a teeny increase in cytosolic calcium. Now, if we then add histamine again, we know the stores are full. We see a large calcium release and transient emptying of the calcium in the ER. Compare this physiological ER emptying with that induced by a SERCA blocker, whether thapsigargan or CPA. And we can quantify all of these things. So this is histamine release. This is the important part here. This is the amount of physiological SOC compared to the SOC induced by thapsigargan here, which is shown here, okay? And compared to calcium release when the stores are full. Okay, so we know the system is behaving. We know we can measure calcium.
Excuse me, just yes. Sorry to interrupt in the middle, but I'm just curious, when the calcium is not going to the ER, so how did this calcium transient went down? Where did that calcium goes to?
Here?
Yeah, before this, like, you know, the first one where the calcium is released from the ER.
Oh, so, this one?
Yes. So how did it go? Where did that calcium?
This is, I mean, the only thing that's left is PMCA and the sodium calcium exchanger at the plasma membrane. Which
but aren't we working with the oocyte? Because in the, you mentioned.
No, this is a HeLa cell now.
Okay. Okay.
These are HeLa cells.
All right. Thank you.
Sorry. Yeah, switch too fast, maybe. Sorry.
No, it's 12 a.m. 1:42 a.m. So
yeah. Oocyte, mammalian cells, they're all the same.
All right. Thank you. Yeah.
All right. So now in the same cell, I'm showing you the mitochondrial signal. To ask the question, what does the mitochondria see from these transients? So during the release phase, there's a huge rise in cytosolic calcium. And you see, coupled to that, an increase in mitochondrial calcium, as you would expect, reported by the mitochondrial cipiatype. So when calcium is high in the cytosol, that's taken up by the mitochondria, and we can see it. Under the conditions where we're activating the cell with thapsigargan, the calcium rise in the cytosol, which is slower, is seen in the mitochondria, but very small, right? Because of the dynamics of the calcium signal, it's not sustained in the cytosol as much, and it's not seen as well, although the ER is much more dramatically depleted here. If we activate store-operated calcium entry under those conditions, that's reported by the mitochondria, as it's coupled also with a large cytosolic calcium increase. Now, what happens under physiological SOC? Calcium rise in the cytosol, ER store depletion, and now when SOC is activated, it does refill the store, but the mitochondria doesn't see anything. Okay, so the calcium flowing in this microdomain is invisible to the mitochondria, although it's very capable of refilling the ER stores, as you see here. And when we release again, the mitochondria sees that beautifully, and we see the transient depletion of the ER. So what that says is the mitochondrion is able to see a large cytosolic calcium rise, as you see here and here, but not calcium flowing through store-operated calcium entry, at least in these cells where the mitochondria we know is deeper into the cell. Okay, so that's perfect. So we know these mitochondria, what kind of calcium signals they're seeing. So now we ask the questions, what happens during tunneling? Right, so during tunneling, calcium flowing through store-operated entry is taken up by circa and re-released into the cytosol. And because of the data from pancreatic acinar cells, we were fully expecting that this tunneling will activate the mitochondria. Mitochondria will be able to see that. So this is our protocol for calcium tunneling in these cells. What we do is we empty the stores with CPA, which is reversible. Remember, for tunneling to work, circa needs to be active. And then we wash out CPA. And now at the same time, when we put the cells in calcium containing, we add histamine. So not only are we allowing calcium flow through SOC, we're opening the IP3 receptor, thus inducing tunneling in the first phase. When you do that, you see how effective tunneling is. This teeny SOC signal, just to remind you, this is the SOC signal that's happening through the in the cytosol. If we look at calcium flowing, again, very reminiscent of what we see in the oocyte. Now is amplified tremendously by the opening of the IP3 receptor. You see a huge calcium rise in the cytosol. What's fascinating, that's completely invisible to the mitochondria. The stores refill, as you would expect. Once the stores are refilled, now if we add histamine again, this is the same exact cell. We release calcium from stores. The mitochondria see that beautifully. And the stores empty transiently and refill, right? So this is cytosolic calcium. The signals are identical during tunneling or during release, pure release. The mitochondria does not see the tunneling calcium signal, but it sees the release signal. So this gives you a feel then on how calcium in the same cell now, with what appears to be the same dynamic based on the reporter you're using in the cytosol. And the vast majority of papers in the literature, that's how people study calcium. They load the cell with a calcium reporter and the cytosol, and that's that. And then they make conclusions based on that calcium signal. But the different reporters are seeing very different signals, although if you just look at this signal, the signals are identical. So how about in the same cells now, looking at a membrane effector, similar to the oocyte? So HeLa cell express calcium-activated potassium channels, not chloride channels. And now if we do patch clamp experiments and empty the stores with CPA and calcium-free, activate SOC by calcium readdition, we can see the activation of calcium-activated potassium channels. Certainly, if we induce calcium release with histamine, we see a very large activation of potassium channels that are calcium activated. And under the tunneling modality, where we empty the stores with CPA, wash CPA out, and then add histamine and calcium to induce tunneling, you see a much slower calcium rise, as you would expect. And that's very effective at activating the calcium-activated potassium channels. And these are quantified here, where you see release is quite effective at activating the calcium-activated potassium channel. If we look at the current peak, release is more effective. If we look at the total charge transfer, tunneling is much more effective in terms of activating the potassium channel, because the potassium channel in this case, although the peak current is not as high, is activated for much longer period of time, inducing more activation of the channel. To confirm these data, we transfected the cells with LCK-GCAM, which is basically a calcium reporter that now localizes to the cell membrane. So it preferentially reports cortical calcium compared to deep calcium in the cytosol. as you would see with, say, Fura or Flow or calcium green or any of the other calcium reporter that are diffusing. And we do these experiments under turf microscopy. So we're only looking at the evanescence plane for these cells, which is 50 nanometers above the cover slip. So we're pretty confident we're looking at cortical calcium and not calcium deep into the cell. If we do this and we quantify the calcium signal reported by LCK-GCAM is quite high during the release phase, as you see with the calcium activated chloride channel. If we activate store operated calcium entry, these are the dynamics of the calcium signaling, and these are the dynamics with tunneling, which basically pretty much explain what we see with the calcium activated chloride channel in terms of potassium channel in terms of amplitude versus total activation as area under the curve. And these are quantified here, the peak with CPA and fabsigargan in response to release and under the tunneling modality in comparison to the area under the curve reported by LCK-GCAM under fabsigargan, CPA, release, and tunneling. So you see the tunneling modality then is very effective at activating long-term signal in the cortex of the cell. So to summarize all of these data, what we think happens in mammalian cells, and for that matter in frog oocytes, once you activate store-operated calcium entry, that signal, calcium flows in this microdomain. It's taken up by Circa. It refills the stores under normal conditions. If under release conditions, if the stores are full and you release calcium from the stores with IP3, you get a huge calcium release from stores that can spread and activate everything. Effectors that are deep into the cell and effectors that are at the cell membrane, in this case, the calcium activated potassium channel. Under store operated calcium entry, this microdomain of calcium is physically very localized, so it doesn't activate the calcium activated potassium channel very well, although it does a little bit because it's close to it. And under tunneling conditions now, what happens is calcium flowing here is taken up into the ER, re-released through the IP3 receptor, much more effectively activating the calcium activated potassium channel. But because the leak through these IP3 receptors is so much larger than the ability of Circa to keep up, that calcium never refills deep to activate deep effectors into the cell, like the mitochondrion in this case. So, Ghanem, how are we doing on time?
Well, I mean, we are a little behind on schedule anyway, so maybe you want to maybe summarize and read.
Yeah. Okay. So I'll skip this part. It's not that critical for the model. So I'll skip all of this. I mean, it's kind of interesting, but not very critical. Okay. So let me close then. All this work was done by an assistant professor in the lab, Raphael Courjare, who really has led this project beginning to end with all these data on both oocytes and mammalian cells. So let me go back here. I'll leave this up and be happy to take any questions.
All right, thank you for very interesting presentations. Time for questions.
Yeah.
So, yeah, go ahead.
Go ahead.
No, I was just going to say, you know, the talk is a little more physiological, but I'm hoping it will generate enough interest that you guys we're going to want to model all of this.
That's why I have a student and I really like the data you presented. I hope you have the data, so I may contact you sometime.
Sure. Yeah.
Yeah, I mean, we have already the model, you know, for the oocyte, so we can make changes to that model and implement these new.
Yeah, we tried to get Ghanem interested, but then he got
Ghanem
very
is
busy.
too busy. He's too big now.
I mean, so At the time when I tried it, I tried to do it very quickly, and I really didn't understand the data very well to succeed. I was just, I had recently moved to a new place, so now I understand the data. But I have actually another interest in it now. So I'll send you a paper that just recently came out in New Run like a week or two ago, and they look at the role of the store operated calcium entry in the neurotransmitter release. But I don't think they explain. So what I believe they think is happening is these microdomains actually are directly affecting the vesicle release. But I mean, just knowing how small, spatially small these microdomains are, I thought that was really unlikely to happen. Although the spines are filled with the ER, but maybe you would know, you would understand it better, because it kind of left some questions open for me.
Right.
So I wonder if the tunneling is playing a role.
Yeah,
There.
I mean, this gets complicated very quickly, Lana, depending on the spatial structure of the cell, the different dynamics. Andreas Goose and colleagues also just modeled this, not tunneling, but the SOC microdomain at the immunological synapse as well. I think the paper is coming out soon. So I think this is really very amenable to modeling now, to ask questions that, I mean, experimentally we can only go so far, and manipulating those things is very hard. I think what's very, very interesting now is how do these things work in the context of the spatial distribution of these different effectors.
Right.
And we have two models. We have the HeLa cell and we have the frog oocyte, where we know physically where these different effectors are sitting. And it would be quite interesting to do something similar to what we've done for calcium signaling remodeling during oocyte maturation, and ask, do we really have all the parameters to explain the data? And that's ideally suited to a mathematical model where you say, if I give you these parameters, do you replicate?
Yeah, so I actually have a question on that, Khaled, and actually also for you, Ghanem, is, from the point of view of modeling, I mean, that's really my concern that, okay, I mean, the experiments have identified some parameters and made some measurements, and there's there's a plausible explanation. But when you do a modeling, that they, basically, if you have too many floating parameters, how useful is the model? I mean, I think that's really a concern with people from the outside. You guys are clearly sold on this. I mean, because this is what you do. But from an outsider point of view, that would really be a concern. And then, for example, one of the things that I was trying to understand here is, no, so I think Khaled's data is very nice. I mean, and it shows clearly the difference between release and tunneling. So I think we can all agree on that. But what is causing it, and whether some kind of modeling can really give insight into it, that's much less clear. For example, I don't know what's known about the buffers, and so the diffusion of calcium will depend also on the concentration of calcium buffers, right? I mean, and things like that. So I wonder. I mean, Khalid, are there some experimental estimates of buffering? Because of course that could affect localization and could explain, compartmentation of buffers could explain a lot of this difference between release and tunneling.
I mean, Shahid, these are very, very good points. And I'm certainly not a modeler, nor am I a mathematician. But the way I think about this is I mean, I kind of think that the amount of parameters is actually not that big, because what we're looking at here, if we look at this slide that I have up now, yes, certainly buffering is critical, but the advantage of looking at this in a model is buffering is not changing under the tunneling, store operated calcium entry or release conditions, right? So that, you know, we can set it and not worry about it. And certainly there's a lot of models in the literature about buffering of calcium and the cytosol, mobile buffers and mobile buffers, how they do this, how they do that. So I mean, we can make it as complicated as we want down the line, but I think initially a very simplistic targeted approach would be the way to go in my mind. So let's take all these parameters that you've mentioned, which are critical, no doubt, and keep them constant, not worry about them. You know, this is what's reported in a typical mammalian cell. We'll use that. The things that are of interest is, we know also from many data in the literature, is the structure of these ER plasma membrane junctions are not changing before and after store depletion. So we don't need to worry about that, except that now calcium is flowing through ORI under these conditions, whereas before it's not.
So I guess what you're saying, what kind of rescues the modelers, so to speak. I mean, I'm sort of putting Ghanem on the spot here, but what kind of rescues the modelers is that there could be an X number of unknown parameters, but because they don't change, you're only worried about the relative, you know, the differences. Exactly. And so yeah, okay, no, that, I mean, that's a valid.
So I mean, there is that, but then another thing is, you know, calcium signaling is probably one of the, you know, the field where a lot of these parameters are very well constrained. You know, the IP3 receptors, the store operated calcium entry, the ORI channels, you know, their kinetics are, and even the buffers, if you consider different buffers, their on and off rates, those things are very nicely constrained by experimental data. So you don't have a lot of room there either. You know, you can just change so much, but there are a lot of constraints that you have to follow. So
there's a large background literature, I guess.
Yes.
This area that helps.
Yeah, this is Dr. Shah. This is pretty established. You know, the buffering constant, the rate constant, everything is available in the literature, and we are using them time to time.
Okay, yeah, but I think that the key point is what Khaled said, that you know, I mean, because
I think. I mean, one critical question that's very difficult to answer experimentally, where the model would be very powerful, is the rate of circa uptake into the ER versus IP3 dependent calcium release. And we've played with that a little bit. I think every IP3 receptor opened can take up to 250 circas at full capacity. Based on the rate of transport. I mean, circa is very inefficient at transporting calcium, or at a very low rate, if you will. So what would be very interesting from the models, and actually what's also very interesting, is a lot of these molecules have been crystallized, or we know their EM structure at the atomic level. So is there, and we know the space of the ER plasma membrane junction, we know its extent, we know all of that. We actually even have estimates of how many ORI channels are in the junction. So the question from the modeling would be, is it realistic to expect, or how many circas would you need in that domain to actually mediate this effectively, mathematically, you know?
So those questions. Question, Khaled. Under physiological, I mean, these are very distinct modalities of calcium signaling, right? I mean, one is very local, one is sort of global, and it's one is sort of semi-local, right? Have you thought about, under physiological conditions, how would you sort of switch, or as per need, switch between one or the other, right? I mean,
yeah.
So also the geometry is going to play a role here, right? So is my assumption correct that the tunneling is mainly because of concentration gradient? It's the driving force is so big that that it sort of stays this semi-local, you get this semi-local spread of calcium signal, whereas, would you agree?
Yeah, no, that's a critical question, Suhita. I think ultimately that's what all this work is heading for. So let me try to explain it with a typical calcium transient that you would see. Now this is the chloride channel. I don't want that. Let's take a calcium release signal like this one.
Right.
So stores are full.
Right.
I add histamine. Calcium goes up in the cytosol.
Right.
One of the effectors sees it very nicely. Actually, both effector in this case see it very nicely because I'm under this modality where now the calcium rises so high, everything sees it, right?
Yeah.
So this is a typical physiological response.
Right.
You know, the cell sees an agonist, calcium is released, and then the signal disappears. Because the agonist disappears, everything goes back to steady state.
Right.
So where does tunneling function here? So you have the release phase, the peak, which is due to calcium release through the IP3 receptor.
Right.
That reaches a peak. Now, you know, we need to expand this to look at it better. But you see there's a plateau here.
Yeah.
Right? That plateau in my mind is the tunneling. Because during that time, now the IP3 receptor is open because there's still some IP3 because of the histamine. There could also be some IP3 because of calcium-dependent activation of PLCs that extend the signal. And then once the IP3 starts to go down, the IP3 receptor shuts down, the stores refill, you close down. Right? You refill the stores, you can see it, you close down. So that, in my mind, physiologically is the window of tunneling.
Right.
If you have more calcium-dependent PLC, if your agonist activates more IP3 receptor release, you can extend the duration of that. And we know very well from the literature that the temporal dynamics of calcium signal can activate very different effectors, right? So I've kind of shown you this under these conditions, where we see differential activation of the mitochondria or the calcium activated potassium channel, but you can let your imagination flow about many other effectors that are differentially affected, and you can then imagine how the duration of the tunneling and the extent of it can lead to very different physiological responses. So what we're trying to do to answer that question specifically, and I didn't have time to go through this, I'll breeze through it very quickly. What we've done here, what we wanted to do is specifically inhibit tunneling and not release or suck, which, if you look at this, is not an easy thing to do, right? Because the effectors are identical. So how do I inhibit tunneling without inhibiting suck or release? And the way we thought about doing it is inhibit circa only at the ER plasma membrane junction, not circa that's found all over.
Okay.
And the way we did that is we used an ER plasma membrane junction marker called mapper. It's well described in literature. And this thing goes specifically to these domains.
Wow.
It doesn't spread throughout the whole cell because it has a STIM1 transmembrane domain, and it has linkers, and it has a polylysine region that binds to PIP2 at the cell membrane. And then all we did is we switched the transmembrane domain with either phospholamban or sarcolipin, which are known to inhibit circa. And the data I was going to show you, I'm not going to go through it now, but basically what it says is we can specifically inhibit circa in the tunneling modality under these conditions. So now we have the tools to answer the question that you asked. And what we're trying to do now is take that to a physiological model and ask what happens if I inhibit tunneling specifically the physiological function.
So we have a couple of questions in that attendees. I want give them a chance. Ridwan, you can go ahead and ask your question.
Yeah, I think, Ghanim,
Yes.
I think we should move on.
Well, you've raised your
Okay.
hand for a
I
while.
have,
All right, we
I
can
have-
take one brief question,
Yeah.
but before we go to that, I just wanted to point out the schedule here. So your talk, Ghanim, is, let's see, is 10:30 to 11:10, and it's already 11:10. So, I think I'm going to moderate your talk, and I think we should dispense with the question and answer session, and we can take one quick question. Radwan.
Yeah, I'll ask this question. Thank you, Professor Khalid. This is interesting. My question is maybe more technical. When you inserted uh, the electrodes into the cell membrane of the egg, did you just puncture that membrane, or did you do
Yeah,
electro?
we just poke it, yeah.
And where is the leakage current from the story? Because I saw the data, it was reported that the peak current was in nanoamp, right?
Yes, correct. I mean, it can go up to microamp in the oocyte.
Could go to micro. The size of the channel is the channel, the sodium channel is what? The diameter.
Oh, this is we're looking at chloride channels in the oocyte. But remember, you have a huge surface area in the frog oocyte. The cell is 1.2 millimeters in diameter, and there are tremendous amount of invaginations at the cell membrane. So the amplitude of the current is very large.
But you took in, for sure, you took in your consideration the leakage current. That recording is just the recording through the channel, not the leakage current, right?
Yes. No, we're sure about that. Yeah.
Thank you.
Okay, great. So I think all other questions should go into the chat, and then the speakers can, address them in the chat or separately. Ghanem, are you ready to go?